Hello, we are continuing to highlight the participants of the virtual research poster competition. Joining me today is Kasey Boger. Hey, Kasey.
Hi, how are you?
Good. So tell us a little bit about yourself.
Yeah, so I'm Kasey. I work under Dr. Blixlager. I've been in his lab. I'm a second year master's student in his lab. And yeah, I did my undergrad at State, and I was working for Dr. Blixlager at the time in the lab, and then just decided to stay on and do a master's. So
Neat. And tell us a little bit about your poster.
yeah, so when I came into the lab, we were primarily working with studying a cell line called IPEC-J2 cells. So this is a small intestinal porcine epithelial cell line. And so we were looking at the cell line and really looking a lot at barrier function. So our lab at the time was really focused on how relevant the cell line is as we look at barrier function. We've had a lot of issues in the past with cell lines really not reflecting a physiologically relevant or even similar measurement of TER, and that's the kind of the trouble we were having with these cells. And so, so when I came in, we really had started looking at, okay, well, how can we either maybe modify or manipulate the media that we're growing these cells in to make them more physiologically relevant in terms of, when you look at so like a small intestinal crypt, you have a variety of cell types. And so we wanted to make sure that, at the cellular level and genetic level, we were seeing that distribution of different types of cells. And then also at a barrier function level, we're seeing, the resistance we would expect across the epithelium. So when you're absorbing different materials, we wanted to make sure that that also was reflective of an actual, intestine of pig. So we started with modifying this media. So we went with it's called a WRN media, which really just focuses on adding in factors that are supposed to increase stem cell populations, proliferative cell populations. And what we found was when we grew these cells in that media, we really saw a decrease in the resistance from growing them in normal standardized media. And that was good. We did see that, and that was promising. So really what we've done since then is just continue to compare the cell line to also primary culture. So we've been taking crypts from neonatal pigs, growing them as a 2D monolayer, and then studying all three of these together. So looking at the cell line, looking at primary culture, and looking at actual tissue, and just saying what different types of cells do we see, what kinds of resistance and barrier do we see, and how relevant is this to an actual intestine. And really it's just optimizing this model so that we can use it further. And so my poster really just talks about mainly the two different types of media that we've used and the results we see. We did some IF on that to look at that closer, just so that we can, as scientists, have the best model and the best optimized model for what we would do. And specifically with our lab, using the pig as a model for, we can take that and use it for large animals, or we can even use that for some kind of human-based studies.
Thanks for sharing, Kasey.
Yeah, thank you.
And take care.
Yeah, you too. Thanks.